Thank you very much for inviting me today. This work happened a little while ago, so I'm sort of catching up and sort of trying to remember all the things we did. It's been a bit of a blur with all the COVID and everything that's gone on, but this was quite an extensive amount of work and a contribution from a number of different people as well. Firstly, I'd like to acknowledge the traditional owners of all the places we did the surveys on. Too many to name because it was quite extensive, right from Gari all the way down to the New South Wales Victoria border. But I'd like to really highlight the work we've done with the Butchulla Land and Sea Rangers on Gari. We've been working with them now for a number of years and really appreciate the input they've had into the work that we've done over the years. So I won't go into sort of the fire side of things. Everyone's really quite aware of the impact of the fires and the extent of the fires over New South Wales, Queensland and Victoria. But the research we had was aimed at looking at the impact of myrtle rust on forest regeneration after wildfire. We were looking at different regions, different forest types where we could, and different fire intensities. So everything from that severe damage where you had full canopy consumption right through to where we just had low level, just the understory affected, just to try and see where myrtle rust was having the biggest level of impact. So when we're looking at the disease, we aim to look at the species that was susceptible to rust. So looking at any particular species, we're just doing walk through transect surveys to start with. But then we started looking at more targeted. Once we've identified a particular species getting affected, want to know what the susceptibility levels were within those species as well, whether it's variable in terms of potential resistance to rust, and the types of regeneration that were being affected. Was it just seedlings, or were we getting significant levels of impact on reshoots as well? So just as a really quick overview, everywhere we surveyed, we found rust, apart from the southern New South Wales areas. We only did one survey in that area. We didn't get into Victoria because the borders were closed, but we only did the one-off survey. Again, it was COVID related. I couldn't get down there. We had to actually pay for someone to go down there and do it independently. And it was before a lot of the rain had come through. So whether we would have a difference if we went back now, I'm not quite sure. There were certainly species there we would consider susceptible. So, you know, we did look hard, but we didn't find any rust at the time. But all the other sites, we saw a really rapid increase in disease. As soon as we started to get that bit of regeneration, a little bit of rainfall returning, rust came in pretty quickly, and the population level of rust really exploded once you start to see those first few dots within sites. We found disease, as I said before, in all fire severity levels. So even in those low impact areas, we were seeing infection occurring on regeneration. Disease, again, on reshoots and seedlings, wasn't discriminating between one or the other. Approximately 36 species we identified in our surveys with some form of impact on rust. A number of those were new records we'd found, never found in the field before. I've got those listed there: Leptospermum speciosum, Melaleuca squamea, Eucalyptus amplifolia, subspecies amplifolia, Eucalyptus pyrocarpa, and then on Gari, the Syncarpia hilii, and I'll talk about that one in a bit more detail. So we saw impact on a range restricted and threatened species, including the Eurymyrtus australis. It was an interesting one. When we first went up into Nightcap National Range, we saw a few individual plants affected, but over time, more and more individuals became infected. And we actually saw, as those individuals started to produce flower and fruit, we saw quite significant levels of infection occurring on the fruit. Rhodamnia rubescens is probably one of the worst hit of those that was regenerating within the fire areas, and I'll go through that in more detail in a minute. But we also saw impacts on common species, so the Melaleuca species and Eucalyptus species as well. So fire and rust on Gari, and this fire was that's on this map here is a little bit later than I guess those 2019. So it was that wildfire that was started by the illegal campfire. When other areas had started to recover from fire and the rain had started to return, Gari was still quite dry, so it was still quite susceptible to fire. And this fire burnt significant amounts of land on Gari. I think it was 80,000 hectares in total from memory, and affected Melaleuca wetlands, wet sclerophyll areas, and rainforest. And we surveyed most of those sites. There is an area on the southern end of the island that was a wildfire sort of in 2019 that we also assessed Melaleuca quinquenervia plots down there. So Satna, it's an interesting one. We'd never seen, in all the years we've been doing surveys on the island, we'd never seen infection occurring on this species. We'd only seen it in the glasshouse. We'd seen seedlings before, we'd seen reshoots occurring before, but post-fire was the first time we'd actually seen levels of infection. And some of the infection levels on individuals were quite severe. The number of seedlings that we saw, so we assessed it 3 different times. We didn't set up permanent plots because we were never quite sure whether we were going back to these sites, again, because of COVID and other restrictions. But at each assessment time, the first time we went there, tiny little seedlings, less than 10 centimetres tall, 20%, or roughly 20% of the seedlings were affected. And again, the second time, similar sort of number. The third time, we started to see almost saplings, I guess, and you can see Tilly in the picture there. So individuals at that height just starting to get infected then. So they'd shown no evidence of getting infected at a younger age. There's no dieback on those plants, but the new shoots are actually getting infected. So would have been interesting to follow this one through a little bit longer than what we did, but we just ran out of time and funds to actually do that. What the impact will be long term, not quite sure, but it's just interesting that we've never seen rust occur on this species until now. The other interesting one, again, on all our surveys, we hadn't seen much infection occurring on the backhouses of myrtifolia. It was only until we saw the fire come through and we started to see infection, but we also saw infection occurring in the unburnt areas. So it's probably an indication that we were seeing a bit of a change in climatic conditions that were driving the disease in the other sites as well. But what was interesting in the burnt areas, we had pretty high levels of infection occurring on reshooting. And in adjacent unburnt areas, there was only 25 or half the amount of individuals that are affected. And the severity of infection was nowhere near in the unburnt site as it was in the burnt sites. As I mentioned, Rhodamnia rubescens was one of the more severely affected. As most of you will know, it's now listed as critically endangered and one of those species we're targeting to try and conserve and capture as much germplasm as possible. And then post-fire regeneration through the Gondwana rainforests, all through Queensland and into New South Wales, down to the Washpool National Park, we found infection at all those sites. It was variable, just because I think of the climatic conditions and the stage of regeneration that was occurring. But we assessed 96 trees across 25 different sites, and 70% of those with susceptible flush had some level of infection on there. We did go back to a number of these sites. We were able to go in. Some of them we were restricted and we couldn't even find the trees again because so many other things had grown up and smothered these things. The rain that occurred had washed out a lot of roads, so we couldn't access some of them. But we were able to get some good data from Washpool National Park. So in assessment 1, there were 52% of individual trees that had dieback. Assessment 2, when we went back there in 2021, 84.44% of trees had some sort of dieback associated with myrtle rust. So there was a big jump. It probably indicates that, you know, these one-off surveys, they tell you a little bit, but they don't tell you the whole story, particularly in terms of that progression of decline and level of decline you might see within individual species. And certainly when we went to other sites, we did pick up new species where we did do repeated assessments. So, you know, myrtle rust is not one of those things I think you can just go in and do a single survey and expect to see all the results. So one of the ones that we, through our transect surveys and our walkthrough surveys, through particularly the coastal heath areas and some of the inland woodland areas, we identified Melaleuca nodosa as being particularly susceptible. Everyone knows that as the prickly leafed paperbark, and it comes in either a heath form or a small tree form. And you can see that in the photos there, the one on the left being the heath, that's in Bundjalung National Park, and the small tree form there in Yarrangully National Park. So what we tried to do with this species is almost look at a north-south distribution. So basically we had plots from Yarrangully all the way down to the central coast of New South Wales. And then we also looked at an east-west transect. So starting Bundjalung, I guess, and then west right out to Banyaba. Unfortunately, Banyaba, we only went to a single time, so we weren't able to get out there and put up more permanent plots. But in most cases, you know, every site we went to, you can find the occasional seedling, but in most cases, those seedlings were either infected or, you know, just not at the stage where we could actually properly assess them. But it was pretty rare to come across seedlings in most of these sites. These are just some images just to share. Those ones are on the left. There's in Banyaba. I think we went to that site, and I'll go through the data in a little bit more detail in a minute. When we went to that site, I think the infection was just starting up. So I don't think we saw the true impact of the disease there. But basically every bit of new flush that was coming onto these plants was getting infected. You get that repeated dieback when the apical leader's taken out, produces new shoots, gets infected again, and repeated infection. We see these burning off of the tips. So this is on the reshoots of some of the small trees there on the left. You can see that typical burnt look. I mean, that's after that glowing yellow symptom that you see. Then in the middle there is just some of the seedlings we came across, and you can see the damage that rust was doing to those seedlings. So unlikely that any of those were going to survive. And on the right there is just your typical heath form, where every single new shoot has been burnt off by rust. So this is typically what you'd see with this particular species when it's unaffected by rust. So this is I don't like to say resistant, but free of disease when we did the assessments, and covered in flowers, absolutely covered in flowers and magnificent bloom. And this is what we see when we see rust infected trees. So you can see individual flowers there, but nowhere near the number you should have. And the high percentage of individuals within these sites looked like this rather than the previous photos. This is just some of the data from the sites. So as I said, Central Coast all the way up to the northern rivers. And we had both the heath form and the small tree form, and we were able to get some sites where we did have different fire intensities as well. You can see there in Nabiac where we did have low fire intensities, the infection levels based on dieback or impact, I should say, looking much lower, obviously in comparison to those where we had either high or severe fire impact. Looking at the small tree form, as I said, the Banyaba results a little bit difficult, so I just don't think we were there at the right time. But in general, there you're looking at the lower fire intensities, the lower the impact that we're seeing on those individuals. This is a plot that we set up on one of the small tree forms in Yarra Gully Nature Reserve, and this was monitoring. We were monitoring infection levels over time. We're aiming for every month. This was work that was funded by the Plant Biosecurity Science Foundation, so it followed on from the work where we did the surveys for ANPC. You can see here that there was initially very low number of tree deaths, but as we went over time, with that repeated infection occurring, we started to see an increased number of deaths. There's a big gap between the last two data points, and this is just basically where the borders shut, and I couldn't get into New South Wales, so we couldn't assess it. So there's probably a little bit of an unknown factor here in terms of what was happening with those trees there. But we're looking at 44.68% of those trees that we had within the plot. I should say this is a 50 tree randomly selected plot, and 44% of those trees were killed over the assessment period. And all those that were killed had disease incidence levels of over 75% of new flush affected at assessment times. 73% of the remaining trees had dieback occurring. So we're looking at probably more deaths over time. And one of the important factors here is that we only had 6% of trees that showed any flower at all. And only one of those trees was what we would consider moderate levels of flowering. We didn't have any that we would consider as being abundant flower. Excuse me. In Bundjalung, we had one plot there in the heath site. We had no deaths recorded within that site, but only 3 trees were actually free of disease during the assessment times. And dieback was occurring on 85% of those trees. Interestingly, in our first year of assessment, we did get 60% of individuals having some flower, but only 5 of those had abundant flower. But when we did the assessment in 2022, only 17% of those individuals had evidence that that flower produced flower or fruit, and only 1 tree was abundant. So there was a decline in flowering rates within those that plot. We also did a one-off assessment. We found another plot a little bit further west within Bundjalung. It's a small tree form, and we assessed 90 trees within that site. 85% of those trees had dieback. 79% of those trees with dieback had dieback in all branches, so it's quite significant. And only 15.56% of the trees had evidence of flower or fruit occurring, with only 4 trees abundant. So big dramatic decline on individuals that potentially are tolerant or resistant to the disease and able to reproduce. The other one that we're obviously concerned about a little bit, and not so much in undisturbed sites from what we've observed, but more in these disturbed sites, is the Melaleuca quinquenervia, or the broadleaf paperbark. Again, we had a range of sites we were looking at here, from Gari to Bundjalung, and we looked at both resprout and seedling regeneration post-fire. These are just some of the sites. So we had some inland lagoon sites around Yarra Gully. We had some river edge sites where they were narrow-stemmed individuals, and then we had some swamp sites where we had quite large diameter trees and quite dense stands as well. This just gives you an idea of the decline rate of these individuals. So once they start reshooting, they start to get infected, and over a 3-month period, all that reshoot was burnt off by rust. And within 6 months, sorry, I just got to take a quick drink. Within 6 months, this tree had actually died, so it was quite rapid decline because of rust. But perfect conditions for rust, obviously, with all that humidity around, and obviously plenty of new flush to drive the disease. And it's just showing you just the different symptoms that we're seeing with that repeated dieback of the reshoots occurring. So this is just some of the results we saw where we could actually compare a little bit of burn intensity. So those ones in Queensland along the Great Sandy National Park area, there was a bit of range of fire severity there, and that's where we saw the lowest level of disease, right through to Yarra Gully, Chatsworth and Bundjalung, where there was severe fire and quite significant levels of trees with dieback associated with myrtle rust infection. Ideally would have had more sites where we had a range of different burns occurring within the one area, but it was just not possible. Most sites, it was either just one level of intensity, and in many of those cases where it had been severe, it was just severe all the way through and burned out the whole canopy. But there was one site in the Great Sandy National Park, which would indicate that the lower the fire intensity, the lower the impact in terms of myrtle rust. And I sort of say this with caution. I don't think this is obviously a thing that's going to fit with all species, but for this species, it does seem to have an influence. But again, we only assess this also over a 2-year period maximum, probably 18-month maximum. So we haven't got long-term plots to sort of see whether this changes over time. But in that high intensity area, we're looking at 87% of trees with some sort of significant dieback occurring in comparison to that around that 40% in the low area. Again, looking, I guess, the progression of decline in these plots that we did have within the Yarra Gully, which is sort of an inland area. It's quite dry, I guess, but it's got some quite large diameter trees within there. 66% of all trees assessed were dead at the final assessment. Not all of that, I don't think, is due to rust. I think some of it's the consequences of fire. The problem is we can't really put in control plots with fungicide within these. They're obviously massive trees, so how do we treat them with fungicide? So what's the normal recovery rate, I guess, with or post-fire with this species? It's a little bit of an unknown. I can't find anything particular in the literature, but if there is information out there, I'd really appreciate any help there. What we did know from those trees that did die, all had some level of infection, and most of them were fairly high. So rust did play a role in the decline of many of them. More concerning, I guess, is the flower and fruiting levels. We only had 8 trees out of the 50 that were producing flower or fruit, and only 4 trees were moderate. There was nothing that we considered to be producing abundant levels of flower. Similarly, we had a couple of plots in Bundjalung, the river plot, which is narrow or small diameter trees versus the swamp plot, which was the large diameter trees. And within the river plot, we had 30% of tree deaths over the period, so by February 2022. Others were in significant decline. And we only had 3 trees that were producing flowers out of the 50. So that, again, is really low, and none of those were producing abundant levels of flowers. It was just 1 or 2 flower buds occurring. Excuse me. In the swamp plot, 52% of trees killed by rust, we think. But yeah, I'm still not convinced. Again, these large trees, I'd still need to understand a bit more about the ecology of the species in relation to fire. But there was, on the majority of those, rust associated with, and quite significant levels of dieback as well. And again, the number of trees, we only saw 12% of trees producing flowers on these. I'd love to go back to these sites. It's just not an option at the moment, but just to look at the recovery. But I know in these Bundjalung, I just drove past there today, and a lot of that large diameter tree area, that area is just not recovering. So it's a lot of those trees just don't seem to be producing any flush. Whether there's seedlings coming up, not 100% sure of that either. And based on what we saw with the recruitment side of things, and I'll go through Yarra Gully first, but it's interesting that the data we got from the New South Wales figures, where we're looking at resistance, 35%, 34% resistance within those Yarra Gully and Bundjalung, were quite similar to what we found when we did glasshouse studies with seedlings from a range of different locations in both New South Wales and Queensland. New South Wales populations were always around that 30% resistance. Queensland populations seem to be a little bit higher. I'm not sure what the reason was behind that. But you can see here in Yarra Gully, as I said, 35% no disease at the point of assessment time. I won't say resistant. And only 9% of what we considered highly susceptible. Here we have these ones that are moderately susceptible, where you might get this infection occurring on the apical shoots, and you get that loss of apical dominance, and you get this branching habit. With repeated infection, you get a smaller leaf. I'm just not sure what will happen with these individuals over time, whether that change in structure is going to significantly impact things over time. You would suggest from our previous data that it's going to impact on flowering, but we just need longer term data to actually determine what's going to happen with these individuals. Again, Bundjalung had a much higher level here, 32%, where we saw between 76 and 100% disease incidence, so quite severe infection occurring. Chatsworth was an interesting one. It's an isolated population. It's west of Bundjalung there. It's sort of in the back of, I guess, some of the inlets of the Clarence River, and it's one little swampy area. We only found 15% of individuals that showed no disease at the time of assessment. So whether that's an indication that sort of population might be at more risk in the future, I'm not 100% sure. What I don't know, and nobody's really been able to tell me, is if we get a change in the population structure so that we're reducing it down to 35% of individuals being resistant and surviving, what does that mean in terms of the genetic diversity of the population, genetic bottlenecking, and what consequences does that have, if any, to the species? I think it's something that needs to be looked at in the future, but I just don't have the answers for that at this point in time. So that was a bit of a rush to try and get through that 15 minutes. So it was probably a little bit over time, but just thank you to ANPC and everyone who worked on the project.